Dr. Lwidiko Mhamilawa, please pardon me for the pronunciation, is a lecturer at the Department of Parasitology and Medical Entomology at MOHAS. As a malaria researcher, he has over 6 years of experience in malaria drug efficacy clinical trials, molecular analysis of malaria drug resistance markers, malaria diagnostic tools, and malaria epidemiology. Some of the publications that Dr. Mhamilawa has been involved in include works that explore novel pragmatic strategies to protect the therapeutic efficacy of artemisinin-based combination therapy against resistance from Plasmodium falciparum, molecular epidemiological works on discerning the genetic interconnectedness of Plasmodium falciparum population between Tanzania mainland and Zanzibar, and using artificial intelligence in mobile microscopy to improve the parasite detection. Dr. Lwidiko has experience in being both frontline worker and consulting in epidemiology during outbreaks of epidemics, such as cholera. He has worked in consultancy works with international organizations such as UNICEF in implementation research, has been an expert trainer in Bill and Melinda Gates capacity building program for malarial leaders. Using his experience in malaria epidemiology and the existing evidence of influence of previous malaria drug resistance, Dr. Mhamilawa is coordinating a study that will explore further the triple artemisinin-based combination therapy strategies in Tanzania. The clinical trials will be conducted in malaria endemic rural Bagamoyo district. So we have somebody with us that is, let's jot our questions and then ask after his presentation. So quickly, I'll just invite the guest. Dr. Lwidiko, you're welcome. And please, if you can help us pronounce your name so that we can get it right. Thank you very much.
Thank you. My name is Lwidiko. Lwidiko.
All right, thank you for that, Dr. Lwidiko. So you have the stage. You can just go on with your presentation. Thank you.
All right, thank you. I think I got a little bit of a technical difficulty. I couldn't hear before. I'd like to get a bit of an introduction of the audience before I start. So, okay, if that was done, can that be done again, please?
Sorry, please, I didn't get that.
I'm saying I'd like to know the audience, the type of people I have in the group, just to get to know who are the participants that I'm presenting to.
Oh, okay. So most of us are Science Cafe members. We're majorly biological scientists, so that are interested in knowing things that are ongoing and how to put ourselves, we're just a community that is based on knowing how to put ourselves out there as young scientists and researchers, especially in the biological sciences. So that's the audience you're speaking to tonight.
Okay. So it's mostly masters and by undergraduate level scientists, students,
yes,
right?
Undergraduate, masters, postgraduate, and PhD postdoc.
Okay. All right. Thank you. I'll have a presentation about a few things that are currently working. I don't know how much you already know about what I do, but I'm a medical scientist researcher from Muhimbili University of Health and Allied Sciences in Tanzania. And I've been working in the field of malaria research for over 6-7 years right now. And within the context of the work that we do, my focus has been in malaria chemotherapeutics. So this is in the context of case management, malaria, and most of the malaria work I've been doing are not in the context of part of the vector. It's been more towards the human parasites, and I will share with you a few of the works that I've already done and a bit of a background of malaria, assuming not most of, I don't know if many of you have a background in in uh, in malaria. So I will, okay, so let me see if I can share my screen. Are you able to see my screen?
Yes, sir. Yes, we can see your screen.
All right, great. So just an overview of what I will be talking to you about today is there is ongoing problem with malaria, and that is development of resistance to the already existing artemisinin-based combination therapy, this is a treatment that is recommended as first-line treatment for people who have uncomplicated malaria. So the idea is, what are different strategies that could be used and tools that could be used to protect the therapeutic efficacy of this drug, but also when it comes to management and care of the patients as well. So the presentation will focus mainly on some of the work that I've already done in Tanzania and some of the work that we'll be doing in the future. And I'll walk you through a bit of a background of malaria for those who don't really know, and I'll present some of the studies that I've done in this and some of the papers that I've done, and some of the future perspectives. So this publication, you can find them online as well, but I'll be breaking them down as we go. So one is really on the molecular, on the diagnostics, so detection of the parasites, which is going to be critical when we are talking about the tools that we need to have to detect the parasites in the human body. And then the other publication is under the management, or what are the strategies that we use to, or as alternative for prolonging making sure that the therapeutic efficacy of this drug is high, and also safety assessment of these drugs, and also as well as some of the epidemiological connectedness of these tools. So yeah, so malaria is a deadly disease. Nigeria is no stranger to this disease, as among the ones that are being hard hit, with more than 400,000 deaths every year, and disproportionately affecting sub-Saharan Africa. There are multiple species of malaria that the parasite that cause malaria. One of the most common one that causes severe malaria is Plasmodium falciparum, and that is the major parasite I'll be discussing with you today. And it reaches a complex life cycle involving three major players. So we know there's a human component, there's a mosquito component, and there's a parasite component. And this life cycle, just an overview, shows you that the mosquito bites you, and then you get to, they inject some of the parasites known as sporozoites. And this is an important step, understanding this life cycle of any parasites, not only just malaria, to get to know what interventions could be done. So once the parasites are injected, this is one of the area that could be used for intervention to come and bring in some vaccines, for example, that are currently being used. This RTSS circumsporozoite vaccine is targeting this stage. This stage, the malaria parasites are very small in amount, maybe 10 to 15 Plasmodium falciparum parasites that are being injected in your body or in the body of a sick child. And then they get to circulate in the liver, they reproduce more, and in other species, when in the liver, they can stay there, they can form hypnozoites. These are important because when they don't get into the circulation, they can stay over for many years. But for Plasmodium falciparum, which causes severe malaria, goes immediately into the normal erythrocytic cycle, which means they get into the red blood cells circulation, and then they invade the red blood cells. They reproduce in these red blood cells, and then they burst to cause the symptoms that you see in malaria. So this is the most dangerous cycle. And then a portion of that, maybe 10% of these parasites will go into the gametocyte stage, which is the sexual stage, and they'll be picked up by a mosquito again to continue the cycle inside the mosquito for reproduction, and then get back to human beings again. So all these three major parts are important for control. So for example, here I said the vaccines work well. Here is where the medicine that treat malaria are working on. And then here is where the medicines that prevent transmission, known as transmission blockers, can work on. Unfortunately, for malaria case management, the medicine that you can take for treatment only works in this stage and hardly work on the gametocyte stage. So someone who has been sick with malaria can be treated for malaria, but will still be able to transmit the mosquito. So more than 3 billion people in the world are at risk of malaria. There has been a decline of malaria cases, but then that has stagnated for very recent World Malaria reports. Even COVID-19 complicated malaria diagnosis and control, among other issues when it comes to global burden. And since 2017, we've been seeing an increase in malaria cases, which has been contrary to reversing the efforts that were done in the previous years, which were causing the decline. So even Sustainable Development Goal number 3, which is sustainable healthy lives, is being affected because of the existence of malaria. And this is the case for Africa. Some of the proportion of cases, we can see more than 5 million cases. We're talking about percent of children under 59 months who are tested positive for rapid malaria diagnosis. You can see areas that have red here in Tanzania is also sort of higher than 5 million. We're talking about even in these countries, West Africa. Some countries in South Africa have less than that. But for Tanzania, for example, the 5% of global malaria deaths occur in Tanzania. And this, I think, is not, and I think it's higher in countries like Nigeria as well. Yeah, so for context, this is the map of Tanzania, and the part of the work that I've been doing will discuss it will be done under area that has almost 5% prevalence, which is going to be around the Pwani region, closer to Dar es Salaam. Right. Again, a bit of a background, malaria parasite detection. 3 major commonly used malaria parasite detection tools are microscopy, rapid diagnostic tests, and nucleic acid amplification test, which is NAAT, which is a molecular method. So with microscopy, you visualize the parasite under the microscope from peripheral blood. Rapid diagnostic test, you just detect the presence of parasite antigen in the blood. And then nucleic acid, you detect the parasite DNA. The tools could be polymerase chain reaction, or it could be also loop mediated isothermal amplification, which is another form of molecular tool. So this graph here is critical to get an understanding of some of the limitations of these malaria parasites detection. And this is another good avenue for research and coming up with novel ideas when it comes to malaria control, is when we count malaria parasites in terms of parasite density, So let's say this is the number of parasites per microliter of blood. This is the time as the infection goes along in terms of months. So an initial after the infection, the parasite's density goes all the way up. It can be more than 100,000 parasites per microliter, but then it starts to, go down in case you get treatment or something like that. But the detection limit we're talking about, if you're using rapid diagnostic tests and normal field microscopy, if you have lower than 100 parasites per microliter, this is sort of the detection limit of parasites. So in a normal situation, someone who's sick would have more than 100 parasites per microliter. We're talking about 1,000, 2,000 parasites per microliter. So this is very useful. Rapid diagnostic tests and malaria microscopy are very good tools for detection of malaria in clinical setting. But once patients start to get lower parasite density, either they have been treated before or they have never been in areas where the parasite density is lower than the 100, you cannot detect parasites using these two tools, which means they will continue to transmit the infection if they're lower than that. So let's say you're talking about a place where it's going to almost towards an elimination of malaria. Most of the people have very low density parasites will be missed when you're screening them using rapid diagnostic test or field microscopy. So other more sensitive tools are needed, especially for elimination settings, such as PCR. But PCRs are complicated to do in a field setting. So other tools like LAMP, which are field friendly, that you could do, I will explain here a little. So the World Health Organization recommends the artemisinin-based combination therapy. So this, just for those who have no idea, the drug that is used for treatment is two combined therapies. One is artemisinin, which is a shorter acting drug, but is very strong. And it should be combined with a longer acting drug, which is also strong, but can stay in the body for a longer time. So artemisinin will be there for for 3 hours half-life, but lumefantrine, for example, partner drug, can take half-life of 5 days, which means it will be in the body for almost 28 days and protect someone from getting reinfected with malaria for at least 28 days. So a partner drug could be another one, any piperaquine or something like that. So present treatment for complicated malaria in Tanzania is artemether-lumefantrine. So I'm sure that you will know what other options there are, but it's twice a day for 3 days, and this is the treatment. So how this artemisinin works, it damages parasites proteins, such as heme-activated endoperoxidase activity, so that the parasite proteins cannot detoxify the heme, which is going to kill it. So, and then the other aspect is inhibition of proteasome activity. So this is, it inhibits the parasite's repair mechanism. So it does the damage of the protein, and then the parasite can want to repair it, but it limits the proteasome activity of the parasite. That means it will not be able to repair its proteins, leading to accumulation of damaged proteins in the parasite itself, and subsequently leading to stress-induced death of the parasite. But then there is this growing concern, huge concern, that artemisinin resistance is developing. And for those who know that there was a previous other medication like chloroquine, sulfadoxine, pyrimethamine, and other previous medication, this parasite is very notorious when it comes to changing itself and causing resistance to the medication. And according to traditional definition of resistance to the drug, it means that the parasite does not die even if you give it the medicine within the safe limits of therapeutic levels. But that definition is slightly different in artemisinin resistance, because in artemisinin resistance we define it a delay in parasite clearance time after giving ACT. ACT, it's artemisinin combination therapy. So this is associated with mutations in the polymorphism of the K13 gene. And now for people who are interested in doing research in this area, there's a lot of publications or areas for research to look into whether or not there are the parasites that are circulating in the community have started to develop this mutation in this gene that could be critical for development of resistance. So the polymorphism in the K13 gene. And we define artemisinin resistance as if you have 100 patients and you give them medication and they have clinical uncomplicated malaria, you give them medicine for the full 3 days, and then after the 3 days, you check the microscopy level if they have parasites or not. If more than 10 patients in that group of 100 have still parasites that you can see them in a microscope, then you would say that there is an alert for artemisinin resistance. So this, you could do clinical trials, you could do national malaria control programs in most countries are required to do this testing for that. But then these parasites, they will be cleared off later on by the partner drug, which could be lumefantrine or any other one. But if they're not cleared, then you would be worried that there is a concern for resistance of the partner drug. So in Tanzania, for example, the day 3 positivity rate in microscopy ranges from 0 to 5%, which is good because we don't have clinical resistance yet. But the molecular markers for the resistance have been detected in countries like Rwanda, some countries in West Africa, some countries in Southern Africa as well. So it's a cause for concern. But then, if you remember, what I mentioned to you before, that the microscope detects the parasites if the density is higher. If the density gets too low, you can detect using PCR positivity. So using PCR on day 3, for some of the studies that have been done in Tanzania, we could see that there is still higher positivity rate, more than 10%. You're talking about 28% to 74%. And this poses a question of whether or not what we're seeing with PCR is real parasites that are still lingering around in the low density, or these are parasites that can be transmitted to another patient, or these are gametocytes, or they're not the sexual parasites, or what are they? So they've been linked to clones, some that have delayed parasite clearance, and sometimes baseline characteristics such as high parasite density at baseline and things like that. And these ones are usually linked to recurrent parasitemia, meaning if you follow up these patients for 28 days, they might end up coming back with malaria if they were on day 3, they were PCR positive. Also, there's some research that shows that there is a correlation with persisting gametocytes and asexual transcripts. Do you still follow me so far?
Yes, we do.
Okay. And I hope the content is clear as well. Very clear. And this, yeah, it's good. You understand? If you have questions, you can put them in the chat and I'll address them as we go. And the other component is how lumefantrine works, which is a partner drug. For example, lumefantrine. This is a quinoline drug, which has structural similarities like with drugs like halofantrin, which tend to have history of causing cardiovascular effects, such as prolongation of the time for depolarization and repolarization of ventricular heart muscles, leading to things such as sudden cardiac arrest. So it is important to, when you're doing alternatives such as what I'm going to share with you shortly, as to assess also whether or not ECG shows any signs of toxicity. And this usually lumefantrine inhibits hemozoin, formation of hemozoin and binds heme, leading to accumulation of heme. Now, heme is toxic to the parasite. The parasite loves to eat the heme, but it detoxifies by converting heme to hemozoin, which is food for that parasite. But then this lumefantrine blocks that conversion and leads to the toxic accumulation of heme, and it dies. For just like how we see in artemisinin that it was associated with K13 or Kelch13 gene for resistance. In lumefantrine, also molecular polymorphism in some of 3 common genes associated with the resistance. So PFMDR1, which is Plasmodium falciparum multi-drug resistance gene 1, and PFCRT, which is chloroquine resistance transporter gene, and mutation in PFMDR1, PFMRP1, which is Plasmodium falciparum multi-drug resistant protein genes. And for those who have interest in molecular genetics, you would understand that the mutations. So for PFMDR1, this resistance for lumefantrine selects for N mutation, N86, which is the most common one, with point mutation, and also 184F, D1246, and PFCRT for K76. So this combination of mutations, if you look at what is in here, for those who probably don't notice, so there's the N mutation and F, and then there's a D here, which is also a combination known as NFD haplotype of the parasites, are able to, were known, some of the research we've done that was shown to be able to withstand almost 15-fold higher lumefantrine blood concentration without the parasites dying. So these are molecular detection. If you see this, you're more likely to say that the parasites are going to be resistant. So one of the works with the reasons that we're trying to do some of this work included, despite having a good efficacy, still there's an increased number of cases in mainland, and Tanzania has the island as well. So the Zanzibar island gets still transmission. So Zanzibar island has lower parasite prevalence, which they are looking towards going towards elimination. But because of the connectedness within this mainland and island, we need to get to know what other ways can we use to detect these low parasites densities. But also there is a continued selection of lumefantrine tolerant haplotypes. And then there is evidence of Plasmodium clones that clear much more slower. And historically, drug resistance has been, happening from Southeast Asia to Africa. But right now we're seeing some of the inborn resistance markers that already have reached in some of the countries in Africa as well. So we need to have tools to study what are the strategies that we can do, and what are the tools for surveillance of this Plasmodium falciparum, and also case management. So need for surveillance and need for case management. Some of the strategies that are being proposed with old tools include using loop-mediated isothermal amplification for parasite detection. This is going to be a game changer for you trying to assess whether or not you can do PCR level results or higher level detection in a field setting. So using this tool for testing these kind of tools for field work, but also to double the duration of artemethyl lumefantrine treatment. Meaning, if the parasites have not cleared by day 3, what if we give more drugs for 3 more days and see whether or not these are going to be clear? This is not uncommon to give medications for 3 to 6 days, and most antibiotics are given from 3 to 6 days. So trying to explore this strategy, because making a new medication until to get WHO certification to be recommended for use, it takes a lot of years, while the parasite still continues to develop resistance real quick. But also adding a single low dose primaquine. Primaquine acts in the gametocyte stage, so this one helps to block the transmission. And moving on, there are 4 studies that I will present to you here that with some of the results. So one was to compare the diagnostic accuracy of LAMP and conventional microscopy using PCR as reference standard, meaning we want to see how well does LAMP perform in comparison to microscopy, while PCR is kept as reference standard to see whether can we detect the same amount of positivity on day 3 of medication, when we use LAMP and PCR. And then another study was to compare the therapeutic efficacy, meaning does it cure people, and how safe is it, to compare between the standard 3 days course vis-a-vis 6 days course of artemether when it comes to PCR determined parasite clearance, and post-treatment prophylaxis, meaning how long does the patient stay parasite-free if you give them standard treatment of 3 days and if you extend the treatment. And also another study is to check how safe is it in terms of electrocardiographic changes that are associated with prolonged artemether treatment, and also to examine genetic relationship between a higher transmission region of mainland Tanzania to lower transmission region of Zanzibar. So this is the publication I was talking about, which is detecting of Plasmodium falciparum by light microscopy, LAMP, and PCR on day 3 of initiation. The primary outcome was the proportion of detectable parasitemia on day 3 by microscopy, LAMP, and PCR. And the secondary outcomes were sensitivity of LAMP with PCR as reference standard. So one last piece of background in the methods. So when you compare PCR and LAMP, there are several key differences. One, with PCR, you need to do something known as thermocycling. Thermocycling meaning you heat the blood sample to denature the proteins, and then you cool it down so that the DNA can be amplified, and then you heat it again, and then you cool it down, and then heat it again, and then you cool it down. That needs a thermocycler, which is a machine which is very expensive and needs very highly skilled people to operate. While when you look at LAMP, you don't need that. You just need one temperature, and then the amplification of the DNA continues in same temperature. That is why it's called isothermal DNA amplification, compared to these other ones which are in thermocycling. Conventional PCR requires a lot of preparation in terms of lysis buffers, some labels, there's a risk of cross-contamination. I know some of you have done PCRs, but in LAMPs, there's ready-to-use tools that don't need too many preparation or mixtures. Many protocols are needed in the PCRs, but in LAMP, it's just one single protocol, just heat, and then you get results, you read them. And internal amplification controls are needed in PCRs, but that's not the case for LAMP. So just for your orientation, we did a study that recruited 256 patients. These are the samples that we got. And we were comparing that and some other parasite samples that had more than 2 parasites per microliter. If you recall, the detection limit of most of the PCRs, including LAMP, is less than 10. But if it's lower than 1 parasite per microliter, the tool will not be able to detect. On the right hand side is only samples that had more than 2 parasites per microliter. This slide. So if you check the positivity rate was very comparable between these two. More than 80. There was 84% detected in LAMP and 84% detected in PCR positivity rate. And these two, there's something known as kappa value. Kappa value is how much these results agree in statistical terms. So the kappa value for this was 91%, right? And this is very good in terms of level of agreement for all the samples generally. But then if you go to, if the parasite density is at least 2 parasites per microliter or higher from the PCR results, the level of agreement gets even higher than that, into 95%. So what this means is using a field-friendly tool to detect parasites in a low parasite density setting, instead of going to do a complicated LAMP PCR, you can do the simple method of LAMP and still get comparable results. And the sensitivity, when it's more than 2 parasites per microliter, is more than 100%. It's 100%. So this is what I had to a conclusion in this study, that the LAMP study is comparable as a diagnostic accuracy tool to PCR, and it's a potential future field-friendly tool for molecular detection of day 3 positivity, or even just detection of positivity in low parasite densities. But then an area for researchers needed to look for what does this day 3 positivity mean by molecular method. Is it parasites? Is it just dead debris? What is it? So it remains to be elucidated. The other aspect, this other publication is already published right now, which is looking at the parasite clearance, the cure rate, and Plasmodium prophylaxis of standard treatment, comparing that with 6 days. And the outcome was proportion of PCR detectable parasitemia on day 5 and day 7 in different arms, and also to assess safety and PCR corrected cure rates and selection of genetic resistance markers that I was talking to you about initially. So this study was a randomized control trial, single-blinded, where we had one arm that received 3 days treatment of alu at lumefantrine, and another arm received 6 days plus single low dose, all these single low dose primaquine, and everybody was observed in therapy, which is DOT, direct blood therapy, and clinical parasitologic assessment was done during follow-up. And this study showed very good results in terms of the difference between the standard treatment and the extended treatment when it comes to side effects or safety profile. There was not any major difference in terms of the number of incidences of safety concerns. So it is as safe as just giving the single standard treatment. But also you could see, if you look at this lumefantrine level concentration, we did pharmacokinetics work, and it showed there's a lot of, which was expected. The black, this is the control arm, and the gray one is intervention arm, where they had more medicine. You could see lumefantrine levels were higher compared to the ones that did not, the ones that received standard treatment, as you look over time. So this is evidence that extended treatment indeed has higher exposure of medicine, and it is still detectable higher in blood levels, which means the parasites that will be available in the blood at day 5, day 6, day 7 will be more exposed to more higher medicine compared to the ones that would not be would receive the standard treatment. This is good evidence to show that in case you're looking at options to increase the concentration of drug in the blood, this was a good alternative. But then, because the study was done in a place where there was no drug resistance yet, so the cure rate was comparable between these two arms on day 5 and 7. There was no statistically significant difference, and this was expected because the medicines still work. So, but then the fact that it gave us a good safety profile, this was useful. But then the one that is most critical is if you look at the survival curve meaning how long does it take before the patients come back with recurrent parasites. So in the control arm, where they receive standard treatment, we had 21 patients came back with parasites. Again, they were sick again. But then in the intervention arm, we had fewer number of patients who came back. We still had a few, but it was fewer. It was 14 patients only, right? And then if you look at how long it took, so this is the survival curve. Every patient starts here at baseline, and you follow them up. The green line is for intervention arm. The blue line is for control arm. This here, it means at 95, how long does it take in terms of time down here for 95% of your patients to stay parasite free? So if you look at the control arm, by, let's say, for 90% of your patients to remain parasite free, by day 35, you had more than 10% of the patients who came back sick. But it took longer, all the way to day 42, for 90% of the patients to remain parasite free, meaning the extended treatment displayed potential for extended protection from recurrent parasitemia of this data. So this is a good trend that we saw in this context. So this was for 35 and this was for day 42. What does this tell us? This tells us that we can look for other options in case we are seeing there is a potential for drug resistance. We can use this extension as a tool. And then we had these NFD mutations, if you remember them. More than 90% of the patients had parasites that had this mutation for PFMDR1. So which means they could have developed resistance. But there's no patients in our study that were shown to have K13 gene, which is gene resistant for artemisinin. So in this study, as I said, the conclusion is that extended treatment could be used for protection of therapeutic efficacy because it was safe, but we could not find any superiority when it comes to comparing the two because it was still ACT sensitive setting. So this also happens when it comes to research. Right, so that's what I wanted to present to you today, and the additional parts we can discuss them as if you have any additional questions. So I'll stop there and I will allow for questions.
Okay, thank you very much. So can we just take our time to appreciate our guest speaker in the comment section? Thank you very much. That was very interesting and educating. Like you broke it down like as if you were speaking to youngsters. So we appreciate that. Please, if you have any questions, you can just raise your hand or drop them in the chat box and I'll allow you to speak or read out the question. It's because of time. If we have any questions, let's quickly drop it.
Right, I don't see questions in the chat. I see. Thank you for the comments. I'm glad that you found it.
Yes, I think some people are still typing their questions if they have.
All right, I'll wait to see.
Unmute your mic if you want to talk so that we can be fast. Okay, well, yes, sure, you can go ahead.
Yeah, thank you. Okay, so thank you very much, Dr. Lwidiko. So these are the questions I have. The first is about the resistance. I think towards the end of the presentation, you're sharing that some of the patients became resistant to had parasites that were resistant. You mentioned the PFMDR genes, and so I basically want to ask whether the resistance was coming from their exposure to the drugs, or perhaps there were maybe this transfer of resistant genes across the parasite. Basically, maybe you could just expatiate on a bit on how resistance in the parasites develops. Then the second question I have is I was you didn't share that part of the presentation, but I was really interested in the aspect you talked about, the the effect that lumefantrine can have on the heart, the cardiac function, because I know over here in Nigeria, and maybe I believe maybe it's also possible in Tanzania, that there are times that maybe people buy this use this malaria medication, and then it's not efficient or it's not strong enough, and then they just finish it, and then they get another medication that is considered more potent. So some persons usually undergo that 6-day exposure with malaria treatment. So I wanted to see if there were no adverse effect on cardiac function with that 6-day exposure to the medication. Thank you.
All right, thank you for those questions. One, when it comes to how the resistance develop, it doesn't develop over one course of time. So what we found was already, if you could, let me just show back this slide. Yes, this one. So if you look at this slide, on day 0, before the patient started to get any medication, when they came in the first time, we had 95% of the patients had this mutation already, had parasites that had that mutation already. Meaning, and this parasites mutation of PFMDR has been existing in circulation, has been increasing in circulation very slowly over the years. So in Tanzania, lumefantrine has been used for more than 10 years. And the very interesting thing about this mutation is the N mutation, N86 mutation, is resistance to lumefantrine, but it is sensitive to chloroquine. So the population of parasites who are resistant to chloroquine has been decreasing because of this selection. So meaning when we checked them at the baseline, it was 95%, and then when they came, all the patients that had recurrent parasitemia, the 35 patients, all of them had this mutation. So this indicates that the population of parasites in the area that we studied it, almost all of them had the molecular mutations that would make them resistant. But then that does not necessarily translate to clinical resistance at all times, right? So having the molecular mutation makes the parasite much more stronger, but does not necessarily mean it won't be cleared, because the beauty of living in a malaria endemic situation, your body also helps the drugs to work. So as you use the drug to kill the parasites, your immunity also kills the parasite. So if the drug reduces a big percent of the parasites, and the immunity can't finish them off. But then that's why we look at the proportion of patients who are still positive, which if it is more than 10%, then you start to be concerned. So if you want to know exactly how these mutations come about, it's a little bit more molecular discussion, but they happen over time because they've been exposed. The last part of the question that you asked was whether there was a difference with lumefantrine when it comes to working in the heart. Here's the results. So just for orientation, when we measure the assessment for that, we look at something known as QT interval, and the QT interval is basically when the heart is pumping, there are different waves that are happening. So different nodes that are happening, and the way the ECG machine works. So the QT interval is the interval between from here to here, where the heart is compressing and decompressing the ventricles. And this is the part where the lumefantrine potentially affects. And you measure that interval in milliseconds. If it's longer than normal, if it's prolonged after medication, then that's a problem because the heart is slowing down. If it's not, then but then in our case, we didn't find none of the patients had prolongation. So threshold for clinical concern in this QT interval is if it is more than 500 milliseconds. And if the change in QTC, meaning if at baseline before giving medication, it was X and then after it is Y, and the difference between the two is more than 60 milliseconds, then you'll be concerned. So there's a threshold for clinical concern. And in this case, none of the patients had more than 500 milliseconds prolongation at day 5 in both arms. And in the control arm, where normal, we had one patient that had more than 60 milliseconds. And in the intervention arm, there was only one patient also with more than 60 milliseconds, meaning that there was no difference in potential causing toxicity in extended treatment or standard treatment between the two. So that is the answer to that question as well.
Thank you very much, sir. Well, I hope that answers your question. If you need more clarification, let us know.
Yeah, I'm fine. Thank you very much.
I think there's a question. Okay. There's a question in the chat box from Nasiruddin. It says, concerning primaquine-induced ROS generation to kill the parasites, how safe is it with ACT usage? Okay, that's just sent it, so you can read it too. Which also induced ROS. And secondly, can antioxidant-based drug or drug candidates be substituted for primaquine in the synergistic studies you did? So I think Wells just sent the question, so you can read it and answer.
Yes, I've seen them. Thank you for resending these questions. This is a very good question, actually, in terms of understanding the role of primaquine. But my concern will be slightly different than inducing RS generation to kill parasites. The primaquine, one of the major problems that primaquine causes is within the RS generation is hemolysis. There are human beings who do not have lactate dehydrogenase, LDH, right? This is lactate, they have a mutation in the body, they don't produce lactate dehydrogenase. This lactate dehydrogenase, it tends to lead to people to get hemolysis. So those dependent hemolysis is a cause of concern, especially for people who have that problem. And in our case, we did not find any, when we're doing safety assessment, there was no patient who had, so not like this G6PD I meant, glucose 6-phosphatidase deficiency, G6PD deficiency. And this enzyme, this G6PD deficiency, is the one that leads to people to break, if they get primaquine, they start to break down the blood, and then they get dose-dependent hemolytic anemia. And 7.5 milligrams, we use 0.25 milligrams to kill the parasites. So this was proven to be good, and it is actually very safe. And in our study was one of the contributors of this information. And right now, in places where there is low parasite density, for where the focus is elimination, blocking transmission, single low dose primaquine is WHO recommended to give to these patients, especially in places where the malaria parasitemia parasite density is very low, for example, in Zanzibar. So the national guideline for treatment in Zanzibar is you give normal ACT, but also you add primaquine. But in mainland, because the density, the prevalence is still high, we don't need to give that because the transmission blocking is not going to happen. And then the antioxidant drugs, or drug candidates be substituted for primaquine in these synergistic studies? Yes, there are some other alternatives to primaquine that could be used, but the safety of them is also a concern as well. But I can't mention off the top of my head what is the best antioxidant candidate that could replace primaquine right now. But the WHO recommended medicine is primaquine. Thank you for this question. I think it's a very thoughtful question. And just to add on, next week, the beginning of May, we are starting a new study that is going to look at triple ACT combination. So instead of using artemether-lumefantrine alone, we're looking at using artemether-lumefantrine and amodiaquine, or artemether-lumefantrine and artesunate amodiaquine, which is known as triple ACT. Or you can look even online, there's things known as TACT studies for triple ACT. And we're doing that clinical trial for more than 400 patients to see whether they're going to behave differently and safety of that. So yeah, so that's my answer to that. Is there any other question, Esther, that I missed? I don't see any question in the chat because I was disconnected.
Yes, I think that was the only question you missed.
All right.
Is there anyone with any other question?
Thank you. One?
Any other questions from anybody? Okay.
Right. So if there's no questions specific related to this, I would just like to give you some other tip if you want on a side when it comes to being ambitious in your careers. I have also a side project that I've been doing, building science centers for children to learn better science, technology, engineering, mathematics subjects. The initiative is called Project Inspire. And this Project Inspire does works. We've built like 2 science centers already in Tanzania. And the main idea is just to inspire children to learn, to be curious, to be inquisitive, and to learn science in a different way than how we normally got the experience to do when we were growing up. So let me see if I can share you this screen. Science center we have in Dar es Salaam. Sorry, yes. I'm not sure if you can see anything. Can you see anything, Esther?
Yes, we can see it. But there's no volume.
Oh yeah. So this is a space where children can come over, experience different exhibits, science exhibits, and learn different science phenomena in a different way, do robotics, do experiments which are hands-on, do coding, do have classes to do also the critical thinking, do problem solving skill sets and things like that. So I can put in the chat our Instagram and LinkedIn. We can connect as well later on to see how this is working. But yeah, so I just wanted to share with you that as well. We can reconnect in social media, LinkedIn, and other places in case you have additional questions or further engagements.
Okay, thank you. We would appreciate the link so we can share it with our members.
Right. I would also appreciate more followers on our Instagram, so that'd be great.
Okay. Thank you very much. So do we have any questions or contribution for the last time before our speaker goes? Okay. Thank you very much, Dr. Lwidiko, for that wonderful presentation. Thank you for your time. Sorry about the network issues. We appreciate the sharing your insights and your work with us. We don't take it for granted. Thank you so, so much.
You're welcome.
Thank you everyone for coming tonight. Really appreciate your questions, your contributions, and your presence. Have a wonderful rest of the week. We'll see you on Saturday. Goodbye, everyone.
Good night, everyone. Bye.
Bye.